next speaker is Ivana Zlatanova. Dr. Zlatanova is a postdoc fellow at UCSF. She holds a PhD in cell biology from University Paris Descartes, where she studied the role of innate and adaptive immunity in post-ischemia, cardiac remodeling, and heart regeneration. Her primary research focused on the role of inflammatory cells in cardiovascular disease, specifically myocardial infarction. And her talk today is unlocking heart regeneration, targeting endothelial enhancer.
Thank you. I would like to thank the organizer to inviting me to talk today. Then I'm sorry, I'm going to talk about a zebrafish, which is completely different from what you hear this afternoon. Then it's going to be a little bit challenging to convince you that the zebrafish is a good model too. Then I'm going to talk about heart regeneration. And specifically about how we targeted endothelial cells in heart regeneration. Then you might think, why endothelial cells? I mean, cardiomyocytes, we know that those are the cells we need to make proliferate to have heart regeneration, but we here are interested in endothelial cells. As you can see here, endothelial cells in gray, representing the vasculature, are almost 35% of all cardiac cells from the heart, which is almost equivalent to cardiomyocytes, which is 39%. And this is just a zebrafish heart with the ventricle, one atrium, and bulbous arteriosus. Then, how do we start being interested in heart regeneration? As we all well know here that myocardial injury is a very common coronary artery disease, and as we can see, this is a central issue for mammals because mammals do not regenerate, and we don't have this capacity. Our self-renewal capacity is around 0.5%, which is very low for us to be able to regenerate after myocardial injury. But if you're looking at the animal kingdom, there's much more interesting models for heart regeneration or for any type of regeneration. As you can see here, the hydra is able to regenerate their entire body. Axolotl can regenerate an entire limb. Even recently, we learned that octopus can regenerate even the entire tentacle. But today, I'm going to more focus about heart regeneration, and one of the models that we are using in our lab is the zebrafish model, which is very well known for heart regeneration. Then, since probably now 22 years to be exact, zebrafish was shown to be efficiently regenerating the heart after ventricle amputation. As you can see here, the zebrafish heart has a ventricle, atrium, and a bulbous arteriosus here. And if you cut almost between 15 to 20% of the ventricle of this heart, it's able to regenerate completely without a scar between 30 to 60 days. Then, very interesting that a lot of cells will participate in this event to regenerate the heart. And one of the cells that we came to be interested in are endothelial cells. Then we can see from a lot of different studies since probably 10 years ago that zebrafish are able to reactivate endothelial cells to promote cardiomyocytes proliferation. We know that endothelial cell can be reactivated from the epicardial side, which will be coronary vasculature, but also endocardium will also participate in this heart regeneration. Then our main focus here was to understand how endothelial cells will participate in heart regeneration. What is the gene regulatory network that will be playing a major role from the endothelial cells to regenerate the cardiomyocytes? Then here we are using this model after injury. Then we resect the heart of the zebrafish, and we compare to the sham animals. Then we were very much interested in understanding if there is any cis-regulatory element in this endothelial cell that will be able to respond to an injury and then to express a specific gene to allow the cardiomyocytes proliferation. Then to answer that question, we start looking at ataxic analysis from our data here generated from zebrafish with apical resection compared to the sham animals. And here we come with 1,000 of pigs with ataxic analysis, and this is where we face the first challenge. Then what do we do with all this bioinformatical data? How we can transform this in vivo analysis? Then that was the first challenge. Then what are we going to do? How are we going to select for putative endothelial enhancers that might have a role in heart regeneration? Then the first thing that we made in the lab is that we create this barcoding-based screening method. Then how this worked is, as I previously shown you, we did an ataxic, we had 1,000 peaks, and we exactly didn't know which one of those might have a potential for heart regeneration or not. Then what we've decided to do is to take those enhancers, we call them putative enhancers here, and to put them in those vectors, TOL2 vectors, with a minimal promoter, a GFP, but also a tag, which is very important here. Then, for example, we screen 10 enhancers at the time. Enhancer 1 will be always associated with tag 3, and enhancer 10 will be associated with tag 12. On the same time, we designed this negative control and a positive control. Negative control is an empty vector, always associated with tag number 1, and a positive control is always associated with tag number 2. In that way, we can co-inject at least 13 enhancers at the time in a single embryo to select for putative enhancers. Then here at 72 hours post-injection, we were able to look at the zebrafish embryos this time and to see a GFP signal. But now you might think she's talking right now in the development, and I've shown you ataxic analysis from regeneration compared to sham injury. Then we know that in regenerating tissues, a lot of development pathways are also activated. Then we assume that there is a commonality, a shared program between development program and regeneration program. And this also allows us to make a better screen in development before trying to understand regeneration. Then, but here, because we injected all those plasmids at the same time, and if we observe some GFP signal, we have no idea where this GFP signal is coming from, which one of those 10 putative enhancers might have an activity or not. Then to answer that, we designed a qPCR strategy here only for the tag elements, as you can see here. Then every enhancer was associated with a different tag. Then we qPCR'd the tag, and we were able to make a conclusion if there was activity or not for that specific enhancer. And if we were convinced of the activity, then we do a single injection here. As you can see, cardiomyocytes are in red, and what you are seeing here in a GFP signal, there are just different patterns of endothelial that we selected from here. Then, with this model, we were able to screen for 480 enhancers, and depending on the pattern that we observed, we were able to characterize like 117 enhancers. This is just a little bit to show you how many different patterns of endothelial enhancer can we observe in the zebrafish. There are plenty of enhancers that are very interesting, but for the purpose of today, I'm going to tell you about one specific enhancer, and I will tell you about a functional in vivo analysis of an enhancer, what we are able to do with a zebrafish enhancer to understand the regeneration. Then, as I was telling you that we chose this enhancer because, of course, we are interested to understand if there is a cis-regulatory element that will be upregulated in injury but not in uninjured zebrafish adult. Then we look at here at the chromatin, and what you can see here in our injured animals, we are seeing that there is an open chromatin for this region compared to our uninjured animal. And it happens that this element here of 700 base pair is localized in the first intron of MMP14B gene. Then we injected this enhancer, this time in a single injection. We raised a stable line, and we just wanted to see if we are really talking about endothelial enhancer. Then the enhancer here is in the GFP. Then you can see the activity of the enhancer. This is the zebrafish during development, and we cross it with a FLK line, which is specific for endothelial cells. As you can see, we have a colocalization between the enhancer and the endothelial cells. But our main focus was to understand if those putative enhancer might have an activation and regeneration. But before talking about regeneration, we wanted to see what's the baseline of this enhancer in an adult zebrafish. As you can see here, in an uninjured zebrafish animal, we are seeing not a lot of the enhancer activity at the basal line. But once we did apical amputation of this transgenic animal, you can really appreciate that after 3 days, even 1 day after injury, here there is a strong reactivation of the enhancer at the injured area. Then here we have an element that is able to drive gene expression, maintain that gene expression throughout regeneration, and temper off while regeneration ends. Then, as you can see, that we quantified this by QPC or by GFP signal, and we are seeing that 30 days when injury and regeneration is done, we do not have any more activity of the regeneration of the enhancer. That because this is an enhancer that will regulate gene expression, we were wondering what gene can be regulated by this enhancer. And because I showed you previously that this was localized in the first intron of the MMP14B gene, we wanted to look if MMP14B will be upregulated after injury in wild-type animals. And what you can see here, that 7 days post-injury, we are seeing that there is a significant upregulation of MMP14B in the injured animals compared to the sham animals. We also wanted to see where this upregulation was, and we did RNA scope analysis here. As you can see, there's a strong upregulation of the signal in the injured area of the zebrafish. But with this, we wanted to do staining here to confirm that the MMP signal that we are seeing in the injured area co-localized with endothelial cells, but more likely also with the enhancer that I previously shown. Then here, what we are looking at is the injured area at 7 days post injury in those animals, and we can see that there's a perfect co-localization between the enhancer, the endothelial line, and the MMP14B. Then, because now we know that MMP14B is upregulated in injury, we wanted to test if MMP14B will have an impact on heart regeneration in zebrafish. Then we were very lucky to find a specific pharmacological inhibition here for MMP14B. And what we've done, we treat wild-type animals with the inhibitor or DMSO. And as you can see here on this graph, we are looking for proliferation markers and cardiomyocyte proliferation. And what we can see here is that we are having strong less proliferation of cardiomyocytes in those animals compared to the control animals. We also did the knockout model for MMP14B, and we are observing the same result, that in our MMP14B knockout animal, we have less cardiomyocytes proliferation than the control. Then knowing this, we were like, okay, now we know that the MMP14B, it's important for cardiomyocytes proliferation and heart regeneration, but do we know if the cis-regulatory element that I previously shown you that is upregulated after injury by itself might be necessary and sufficient for heart regeneration? But the first question that we were wondering here is this a really bona fide MMP14B enhancer? Then the first thing that we've done, we did a knockout of this enhancer, and then we check for the MMP14B expression. As you can see here, at 7 days post-injury, there is a strong dysregulation of the MMP14B signal compared to the wild-type animal. We also look at the RNA scope analysis, and we are seeing that we have a less signal in our MMP14B enhancer knockout animal compared to the wild-type animals. But we wanted to see if this is sufficient and necessary for heart regeneration. And then what we've done is the same experiment. We'll look at 7 days post-injury at the proliferation marker, specifically proliferation for cardiomyocytes. And what we are seeing here is that in our knockout animal for the enhancer, we have less cardiomyocytes proliferation. On the other side, we also wanted to look at the scar and see if that's might be affected also. And what we are seeing here is that 30 days after injury, we have more deposition of scar compared to the wild-type animals. Then with this, I showing you until now that MMP14B is a bona fide enhancer of MMP14 gene. It's necessary and sufficient for cardiomyocytes and also scar resolution. But the next challenge that we wanted to address here was to understand and to see how this enhancer might be regulated. Then here, I previously shown you this technique, the barcoding technique. Then I'm going to show you the same. We are going to use the same technique, but with a different purpose now. Then now the purpose is to dissect the enhancer. Then previously, we used it to discover the enhancers, but now we are trying to understand how we can use in an unbiased way to identify a transcriptional factor that might be responsible for the enhancer's activity. Then what we have done here is that we use our enhancer of interest, which is MMP14B, and we decide to do deletion to create a mutation for this enhancer in order to dilate, take out the transcriptional factor binding site that might be responsible for the activation of the enhancer. Then we co-inject all those elements together, and by qPCR, like I previously told you, we are seeing that in the region number 9, we are observing really a decreased activity of the enhancer compared to the wild-type enhancer. Then we wanted to check in the zebrafish embryo what's happening with the activity of the enhancer, and what we have done here is that we used our wild-type enhancer and the enhancer lacking the region number 9. And as you can see here, during development, we don't see much of the endothelial activity when we knock out this specific region of the enhancer. But when we wanted to have a close look at what's in that region, what are the transcriptional factors that might be responsible for the activity, then we look a little bit closer to this region, and what we found here by bioinformatic analysis is that as a perfect transcription factor binding site for TEAD. Then the next step was to do point mutation for this transcriptional factor binding site. And here, after doing that, we are observing that we are also losing the activity of the enhancer compared to the wild type. But our main focus was to understand if this will be affecting heart regeneration, if we're going to lose the activity of the enhancer during heart regeneration or not. As you can see here, when we are mutating the TEAD transcriptional factor site in the MMP14B enhancer, we have no activation anymore at the injury compared to the wild-type animal. Then what I showed you for MMP14B enhancer summary here is that this is an MMP14 bona fide enhancer. It's bound by TEAD, which will activate at MMP14B. MMP14B will, I haven't shown you today, but this was just recently published, that is going to go and activate agrin, and agrin will participate in cardiomyocyte proliferation. But then you might think, why she's telling us about the zebrafish and how this can help the heart to regenerate, because this whole story was to understand how we can use a zebrafish model, which we very know well that is regenerating, in a model like the mammals that do not do regeneration. Then the first element of response here, for us at least, was to look at the transcriptional machinery. When what we have done here is that we took the zebrafish enhancer, and this time we put it in a mouse, and we wanted to see if this enhancer will be able to be reactive in a different model than the ones that I previously showed. Then here we inject mouse, and we raised a stable line, and we were very excited to see if we will be able to see anything. And it came up that the zebrafish enhancer was definitely able to be reactivated in the mouse embryo, and more almost exactly like the zebrafish embryo, the vasculature was exactly activated like the zebrafish. Then with this, we are learning that the transcriptional machinery is conserved between those vertebrates. But once again, the main purpose of this was to understand if we can use this in regeneration or in a model where we do heart injury. Then what we have done is that we raised those mice in a stable line, and that we did myocardial injury this time in those mice, and we look for the enhancer activation. Then what we have done here is this is the sham animals. Those are the myocardial injured animals, and then you can see there is a strong reactivation at the injury site after myocardial injury. Then this was very interesting for us, and this started for us a new project now to understand if we can use those enhancer in a manner to help a mammalian heart to repair at least or to regenerate. Then what I've shown you today is a story about this enhancer specifically, the MMP14B enhancer, which is a coronary vasculature specific enhancer. But we are also working in the lab right now about endocardial specific enhancer. Then you can see that with this kind of a method, we are able to identify different pattern of endothelial enhancers, and they are all very specific and injury responsive for now. Then the idea behind is to use those small elements, very specific, to direct gene expression and specifically regenerative factors at a spatiotemporal manner that we can direct heart regeneration or reparation in adult mice. Then the idea is to use AAV vectors for the future, this is in development, to understand and also to upregulate a regenerative factor in a heart that it's not regulated, that is not regeneration. And with that, I would like to thank all my lab, and especially the Black Lab, and all the collaborators for this work. And thank you.
Thanks, Doctor. Thanks for a great talk. Any questions?
Really nice talk. Thank you. I am curious about when you mentioned the cardiomyocyte proliferation. Do you know or have some idea if this is cardiomyocyte division from the cardiomyocyte itself, or it's actually coming from the progenitor cells that they are before the cardiomyocytes to happen?
Then from now, what is shown in the literature very well, for now there is no contradiction, which is rare. The proliferation comes from the pre-existing cardiomyocytes already that are next to the injured area.
Nice.
Yeah.
Thank you. Very nice talk. And yeah, you mentioned when you did the enhancer part knockout, right? So I want to ask how much database you delayed from this cell line?
How much?
Yeah, how much?
For the knockout enhancer, this is a 354 base pair.
Okay, so based on this cell line, I'm curious about that because of the chromatin structure.
Yes.
Did you detect this enhancer deletion influence the neighbor gene's expression? If there are other genes responsible for this dysfunction?
Yeah, that probably might be happening, but we haven't checked if there is any other genes that are affected by this deletion. We only check about MMP14B because it's an intronic enhancer.
Okay, thank you.
I have a question. Like endothelial enhancer promote heart regeneration. Is there any? What if like over activation? Is there any side effect for the heart regeneration?
There is any side effect?
Yeah, if the endothelial enhancer is too over activated.
If I understand well, if there is any side effect,
yeah,
if an endothelial enhancer is reactivating upon regeneration. We haven't seen that, at least in the zebrafish for sure, because this is a very good model for regeneration anywhere. Whatever you do, it will regenerate. Then no, we haven't observed that.
Thank you. Any other questions?
Have you checked the secreting factor from the MMP14B positive cells?
Have I checked what?
The secreting factor, it seems the regenerate collateral vessel seems induce cardiomyocyte proliferation, like regeneration.
Yeah. Then what we have dissected really well, specifically in the mice, was to understand how MMP14B will promote cardiomyocyte proliferation. And because MMP14B is a metalloproteinase, which is going to degrade collagen matrix, and the idea and what we have checked in mice is that is able to upregulate agrin availability, and agrin through YAP and TAD hippo signaling will be able to promote cardiomyocyte proliferation, which was already very well demonstrated in the literature that agrin is a pro-regenerative molecules. But our major discovery here was that MMP14B is activating or it's liberating agrin into the extracellular matrix to allow for this to happen to the cardiomyocytes.
Okay, thanks.
Thanks again, Dr. Zlatanova.